Good evening to everyone. Welcome to BIREN'S Academy. तो आम प्रीवियस सेल डिवीजन, सेल साइकिल, प्लाज्मा मेम्ब्रेन स्ट्रक्चर एंड फंक्शन विथ साइटोस्केलेटन स्ट्रक्चर एंड फंक्शन आम किछटा क्वेश्चन डिस्कस करथिले। आज आम बायोकेमिस्ट्री किछटा क्वेश्चन डिस्कसन करबा। इथरे फिल इन द ब्लैंक्स भी रखी और मल्टीपल चॉइस क्वेश्चंस अच्छी, तो ये सब प्रकार क्वेश्चन आम डिस्कस करबा। ठीक अच्छे, सो लेट्स स्टार्ट। तो फर्स्ट पचारे आमर व्हिच माइटोकॉन्ड्रियल मेम्ब्रेन हैज व्हिच हाउ मेनी टाइप ऑफ शटल्स फॉर ट्रांसपोर्टिंग व्हिच टाइप ऑफ कोएंजाइम ऑफ साइटोप्लाज्म प्रोड्यूस्ड ड्यूरिंग ग्लाइकोलाइसिस। दैट मीन्स सॉरी एक सेकेंड, that means this is inner mitochondrial membrane has two shuttles। आम जाचे आमर कौन रही ना शटल सिस्टम दुईटा टाइप रही। सो ट्रांसपोर्टिंग NADH ऑफ साइटोप्लाज्म प्रोड्यूस ड्यूरिंग ग्लाइकोलाइसिस। तो आम जाचे शटल सिस्टम जोटा कि ग्लिसरॉल फॉस्फेट शटल सिस्टम एंड मैलेट एस्पार्टेट शटल सिस्टम। ठीक अच्छे। अच्छा, then asla next question, which shuttle operates in cells of skeletal muscle and brain, that is glycerol phosphate। Glycerol phosphate। Then which shuttle operates in cell of liver, kidney, heart, this is malate aspartate। ये है skeletal muscle and brain और malate aspartate asla liver, kidney, heart। There occur about 12 types of isoforms of aquaporine। So aquaporine केते टाइप रा में पाइबा 12 टाइप। Then example of unipolar solute system। So unipolar जानिचे, antipolar जानिचे, sympolar जानिचे। ठीक अछि। So unipolar solute carrier is that is GLUT, GLUT2। Then number 6, this is symports transports two or more molecule in the same direction। Same direction, ठीक अच्छी, opposite direction। Then antipoters are called exchangers and or we can say that counter transporter। V type H plus ATPase occurs in membrane of endosomes and lysosome। H plus ATPase occurs in membrane of endosome and lysosome। Then exocytosis जहाँ को आमे कहूचे constitutive secretion or exocytosis occurs in plasma। Then न्यूरॉन्स रे, एंडोक्राइन सेल रे एंड पैनक्रियाटिक एक्टिनर सेल रे आम ये जोटा पाइबा दैट इज रेग्युलेटेड सिक्रेशन। रेग्युलेटेड सिक्रेशन। सो रेग्युलेटेड सिक्रेशन occurs in neuron, endocrine cells। Then constitutive secretion occurs in plasma secreting immunoglobulins। The next, oxidation and reduction always occur together। ATP synthesis occurs in chloroplast and mitochondria। The role played by ATP in biochemical reaction is that of an energy donor substance or a coenzyme। Energy donor substance, role of ATP एमती आसब reducing agent, coenzyme, energy donor substance or a coenzyme। So an energy donor substance or a coenzyme। Then next, so this is about gluconeogenesis। So gluconeogenesis कौन? Gluconeogenesis कहले आम एटा कहूचे फॉर्मेशन ऑफ ग्लूकोज और ग्लाइकोजन। तो ये जो आसीबो कहाटू आसीबो ना फ्रॉम नॉन कार्बोहाइड्रेट सोर्सेस। So gluconeogenesis is gluconeogenesis does not occur in adipose tissue। एटा मन रखिबा adipose tissue रे हुए नाही। काहे कि हुए नाही ना ड्यू टू लैक ऑफ fructose 1,6 डाइफॉस्फेट डाइफॉस्फेट फिल इन द ब्लैंक्स मु एथि डिस्कस करूँ एथु आम बहुत गुड़ाक वेरिएशन आम पाइबा क्वेश्चन जमी आम ये जानले एडिपोज टिश्यू रे हुए ना आ कौ एंजाइम हुए ना फ्रुक्टोज 1,6 डाइफॉस्फेट देन ग्लाइकोजेनेसिस ग्लाइकोजेनेसिस इज एन एनाबॉलिक एक्टिविटी ब्रेकडाउन सॉरी एनाबॉलिक एक्टिविटीज माने सिंथेसिस जेनेसिस अच्छी तो सिंथेसिस then glycogenolysis lysis breakdown glycogenolysis in liver is stimulated by two hormones, that is one is epinephrine and another one is glucagon. Glycogenolysis in liver is stimulated by two hormones, epinephrine and glucagon. Then during glycogenesis, the step of branching glycogenesis involves two enzymes, that is dextranase called branching and synthetase. The name of the protein that has been identified to serve as a primer for glycogen synthesis is, that is glycogenin. The substrate level phosphorylation in Krebs cycle is catalyzed by the enzyme succinate thiokinase. So glycolysis reduces 2 molecules of NAD for every glucose molecule processed. Most enzymes of Krebs cycles are present in mitochondrial matrix. Which of these conversion in Krebs cycle indicates substrate level phosphorylation? This is succinic acid to malic acid. Gluconeogenesis is number 4, that is B, the formation of glucose or glycogen from non-carbohydrate resources. Then next. Triglycerol are highly concentrated form of energy. Triacylglycerols are transported from intestine as chylomicrons to liver, muscle and heart. Number 3, carnitine carrier system. Carnitine carrier system operates to transport activated fatty acids from cytosol to the mitochondria. Alpha oxidation of fatty acids occur in plants only. PBD or paroxysome biogenesis disorder causes death of human infants within 1 year after their birth. PBD causes which type of syndrome? That is Zellweger. Acyl carrier protein ACP has 77 amino acids. FAS complex stands for fatty acid synthase. Triacylglycerols are non-polar and hydrophobic in nature. Presence of ketonic bodies in urine is called as ketonuria. Oxidizing machinery of beta oxidation of fatty acids is located in mitochondria. Oxidizing machinery of beta oxidation of fatty acid. Then hormone sensitive lipase activity is inhibited by the hormone insulin. Hormone sensitive lipase activity. Then the two final products in the beta oxidation of odd chain fatty acids are acetyl coenzyme A and propionyl coenzyme A. two final products in the beta oxidation of odd chain fatty acids are beta chain oxidation, odd chain, that is acetyl coenzyme A and propionyl coenzyme A. Oxidative deamination of glutamic acid produces alpha ketoglutaric acid. Alpha ketoglutaric acid. एटा भलो कि मन रखिबो. Oxidative deamination of glutamic acid produces alpha ketoglutaric acid. Alpha ketoglutaric acid deamination, oxidative deamination हेले. Then transamination of glutamic acid to pyruvic acid. Glutamic acid ru pyruvic acid हे जिवो. तो कौन कौन बनेबो? Alpha ketoglutarate and 2-alanine. चारटा क्वेश्चन अच्छे, हाँ 9टा. 2-alanine. Pyridoxal phosphate enzyme is derivative of vitamin B6. बहुत इंपोर्टेंट अच्छे एटा टॉपिक टा. Pyridoxal phosphate enzyme. Pyridoxal phosphate enzyme is derivative of vitamin B6. Urea cycle involves ornithine and citrulline amino acid. Ornithine and citrulline amino acid. The next question. The coenzyme that participates in transamination reaction is pyridoxal phosphate. Pyridoxal phosphates. The most important enzyme involved in oxidative deamination is glutamate dehydrogenase. Oxidative deamination पाई glutamate dehydrogenase. और transamination पाई pyridoxal phosphatase. Pyridoxal phosphatase or phosphate, pyridoxal phosphate. Then that a coenzyme na, so pyridoxal phosphate, that is also known as PLP, pyridoxal phosphate. Then N-acetyl glutamate is required for the activation of enzyme, that is carbamoyl phosphate synthase 1. Carbamoyl phosphate synthase 1. N-acetyl glutamate is required for the activation of enzyme carbamoyl phosphate synthase 1. During oxidative deamination, amino acid glutamate serves as collection center for the amino groups. Then glutamine is the storehouse of ammonia in human body. ये सब क्वेश्चन गुड़ा का मोस्ट इंपोर्टेंट क्वेश्चन. Glutamine is the storehouse of ammonia in human body. आउ थोड़े रिपीट कर दूँगी. Oxidative deamination of glutamic acid produces alpha ketoglutaric acid. Transamination of glutamic acid to pyruvic acid produces alpha ketoglutarate and 2-alanine. Pyridoxal phosphate enzyme. Phosphate enzyme is derivative of vitamin B6. Urea cycle involves ornithine and citrulline amino acid. The coenzyme that participates in transamination reaction, that is PLP, pyridoxal phosphate. The most important enzyme involved in oxidative deamination is glutamate dehydrogenase. N-acetyl glutamate is required for the activation of the enzyme carbamoyl phosphate synthase 1. During oxidative deamination, amino acid glutamate serves as collection center for the amino groups. Glutamine is the storehouse of ammonia in human body. Then number 1, the amino acid that does not participate in transamination, that is lysine. Transamination le participate करे नहीं. The amino acids that are said to be ketogenic when the carbon skeleton is finally degraded to acetyl coenzyme. The amino acids are said to be ketogenic when the carbon skeleton is finally degraded to acetyl coenzyme A. The synthesis of urea occurs in liver. Storage form of high energy compound in vertebrates is phosphoarginine. Phosphoarginine. Storage form of high energy compound in vertebrates. A more negative redox potential represents a greater tendency to lose electrons. Negative redox potential. The electron transport chain is located in inner mitochondrial membrane. The component of electron transport chain which can directly react with molecular oxygen, that is cytochrome oxidase, that is cytochrome A plus A3. Component of electron transport chain which can directly react with molecular oxygen. The site of ETC inhibited by cyanide, that is also cytochrome A plus A3. The bonds responsible for a majority of high energy compounds are phosphoanhydride bond. Name the compound with the greatest standard free energy. Number 1, that is phosphoenol pyruvate. Greatest standard free energy, that is phosphoenol pyruvate. One of the following components of ETC possess isoprenoid units, that is option A, coenzyme A. Isoprenoid unit, that is coenzyme A. The P:O ratio for the oxidation of FADH2 is 2. Inner mitochondrial membrane is impermeable to all of them. Hydrogen ion, hydroxide ion, potassium ion, so all of them. ATP synthase activity is associated with the mitochondrial enzyme complex, that is V, mane complex 5. ATP synthase. Iron containing compounds that act as hydrogen acceptor in respiratory chain are cytochrome. Oxidation of extra mitochondrial NADH is mediated by substrate subtle or subtle system. So subtle system रा काम कौन ना extra mitochondrial NADH जहाँ रहिबो तार oxidation. For every cytosolic NADH, the malate aspect subtle yields how many ATP? That is 2.5. And whereas the glycerophosphate subtle system yields 1.5 ATP. Malate aspartate 2.5, glycerophosphate 1.5. Which is the more universal NADH subtle system? That is malate aspartate subtle system. Malate aspartate subtle system. Malate aspartate subtle delivers the reducing equivalents of cytosolic NADH to complex 1, whereas the glycerophosphate subtle delivers to complex 3 of respiratory chain. Which is the irreversible NADH subtle? That is glycerophosphate subtle. Then which subtle does not involve membrane transporter system? That is glycerol 3-phosphate. Two conformational states of carrier protein are ping pong. Hydrophilic transmembrane protein channels responsible for rapid diffusion of water into and out of the cells are called as aquaporins. Hydrophilic transmembrane protein channels responsible for rapid diffusion of water into and out of the cell called as aquaporin. Membrane shuttles that increase the permeability of specific ions are called as ionophores. The potassium carrying ionophore is called valinomycin. While the Na plus carrying ionophore is called as monensin. Sodium carrying ionophore yella valinomycin ionophore. So ionophore kona membrane shuttle that increase the permeability of specific ions. Then number 5, lipase move phospholipid from outer to cytosolic leaflet, while flopages move phospholipids in reverse direction. From outer to cytosolic leaflet, lipase, और reverse direction ले flopage. Number 6, K plus channels are the largest of the voltage gated ion channel. MDR and CFTR are the common examples of A, B, C transporters, which provide the energy for secondary active transport, that is ion gradients. The transport process by which substances move into one side of the cell across then undergo exocytosis on the opposite side of the cell is called transcytosis. Transport process by which substances move into one side of the cell across then undergo exocytosis on the opposite side of the cell, that is known as transcytosis. The study of the energy changes in biochemical reactions is called as bioenergetics. The amount of energy in a system that is available for doing work is called as free energy change. And is symbolized as del G. Living cells are essentially isothermic system and use chemical energy to power living process. When del G is negative, the reaction is exergonic. In case of thermodynamically unfavorable reaction, the del G is positive. ATP contains 2 phosphoanhydride bonds and 1 phosphate ester bonds. दुईटा phosphoanhydride और गोटे है phosphate ester. The standard free energy of hydrolysis for ATP is minus 30 kilojoule per mole, while that of glucose 6-phosphate is minus 13.8 kilojoule per mole. For most enzymatic reactions that involve ATP as phosphoryl group donor, the 2 substrate is magnesium the cyclic AMP, a common second messenger formed from ATP in a reaction catalyzed by adenylate cyclase. Phosphoryl group transfer potential of ATP for on-catalyzed reaction is 200 to 400 kilojoule per mole. The normal intracellular concentration of ATP is 5 to 10 So say 5 to 10 millimolar, while AMP is less than 0.1 millimolar. Then the coupling of ADP entry into the mitochondrial matrix to the ATP exit is mediated by ADP translocase, which constitutes 14% of IMM protein. So there is two example of enzymes containing zinc, alcohol dehydrogenase and carbonic anhydrase. Then name the cofactor. Iron and copper, जे कि cytochrome oxidase by cofactor. Which vitamins act as coenzymes by itself? Vitamin C and vitamin K. Which coenzyme serves the role of acyl group carrier? Coenzyme A. The non-protein part of holoenzyme is called as cofactor. Non-protein part है cofactor. The term cofactor collectively includes metal ions and coenzymes. Apoenzymes, when tightly bound to inorganic metal ions, are called as metalloenzymes. In metal activated enzymes, metal ions are loosely bound to apoenzymes. Then number 5. Which coenzyme was the first coenzyme discovered in 1906? That is nicotinamide. Act as a carrier of hydride ions in redox reaction. Enzymes that utilize flavin coenzymes are called as flavoenzymes. Nicotinamide nucleotides NAD and NADP readily move from one enzyme to another, hence called coenzymes. The flavin nucleotide are usually tightly bound to the enzyme and serve as prosthetic group. Dehydrogenases recognize soluble electron carriers like NADH and NADPH as their coenzymes. NADPH. NAD plus and NADPH represent the reducing power of a cell. The inactive precursors of enzymes are called zymogens. Number 2, phosphorylation and partial proteolysis are frequently employed covalent modification. The rate of enzyme synthesis at the genetic level can be adjusted by the activation of transcription factor. Many human enzymes are regulated by phosphorylation, dephosphorylation reactions catalyzed by protein kinases and protein phosphatases. Which is the most sensitive index of a cell's energy status, ATP or AMP? Ratio. Glycolysis harvests chemical energy by oxidizing glucose to pyruvate. The major rate determining step in glycolysis is phosphofructokinase 1. The predominant hexokinase isoenzyme of liver is glucokinase or hexokinase. And type 4. Breakdown of carbohydrates in physiological conditions yield 4 kilocalorie per gram energy. Glycogen stored in muscles is not available for regulating blood sugar. In RBCs, glycolysis leads to the production of lactate as the end product. The special glycolytic pathway in RBCs is called as Rappaport-Lewering cycle. The conversion of glucose into ethanol is called as alcohol fermentation. Increased aerobic glycolysis in cancer cells is called as Warburg effect. The final electron acceptor in lactic acid fermentation is pyruvate. In alcohol fermentation, NADH donates its electron to acetaldehyde. Which is the final common pathway for the oxidation of fuel molecules? The TCA cycle. Most fuel molecules enter the TCA cycle as acetyl coenzyme A. In eukaryotes, TCA cycle occurs in mitochondria, while in bacteria it occurs in cytosol. The citric acid cycle involves 2 decarboxylation reaction and 4 oxidation reaction. The citric acid cycle is serving in both catabolism and anabolism, hence it is amphibolic pathway. Alpha ketoglutarate is the only 5-carbon metabolic intermediate of citric acid cycle. The pentose phosphate pathway generates NADPH and ribose phosphates. Reactions of the pentose phosphate pathway occurs in the cytosol. The reactions of oxidative phase of pentose phosphate pathway generate NADPH, while the non-oxidative reaction of the pathway generate pentose phosphates. Pentose phosphate pathway and glycolysis are linked by the enzyme transketolase and transaldolase. The major rate determining step in PP pathway is G6-PD. synthesis of new glucose or glycogen from non-carbohydrate is called as gluconeogenesis. Seven out of 10 enzymatic reactions of gluconeogenesis are the reverse of glycolytic reactions. In the first bypass reaction in gluconeogenesis, pyruvate is converted into phosphoenol pyruvate. Number 4. Pyruvate carboxylase is the first regulatory enzyme in the gluconeogenic pathway. All amino acids are glucogenic except leucine and lysine, which produced by active skeletal muscle is a dead end in glycolysis, lactate, and is a major precursor for gluconeogenesis. Propionate is a major glucogenic precursor in ruminants. A high ratio of glucagon or insulin stimulates gluconeogenesis. आम जरूरी ग्लूकागन और इंसुलिन रेशियो बढ़ी जदि ग्लूकोनियोजेनेसिस को स्टिमुलेट करियो. Activation of fatty acid is catalyzed by the enzyme acyl coenzyme A synthetase. Fatty acyl coenzyme A with 14 or more carbon enters mitochondrial matrix through a three-step process called carnitine shuttle. Mammalian tissues oxidize fatty acids primarily by the beta oxidation pathway, which primary occurs in mitochondria. Beta oxidation proper involves a recurring sequence of 4 reactions. Net ATP yield from one molecule of palmitic acid is 106. Oxidation of odd number fatty acids produce propionyl coenzyme A along with acetyl coenzyme A. Then mitochondrial matrix is the major site of beta oxidation in animal cell, while in plant cell it is peroxisomes. Phytanic acid obtained from dairy products and green vegetables is oxidized in peroxisomes through alpha oxidation pathway. Refsum's disease is caused due to lack of enzymes of alpha oxidation, resulting in accumulation of phytanic acid in blood. Fatty acids are transported in blood by the carrier albumin, which is the building block of fatty acid, that is acetyl coenzyme A. In most organisms, fatty acid synthesis occurs in cytosol, but in plants it takes place in chloroplast. The key enzyme in fatty acid synthesis is acetyl coenzyme A carboxylase. The coenzymes required by the acetyl coenzyme A carboxylase are biotin and ATP. The coenzymes required by the acetyl coenzyme A carboxylase, biotin and ATP. The fatty acid synthase complex consists of 2 subunits, and each subunit contains 7 enzymes organized into 3 domains. Palmitic acid is the principal product of fatty acid synthase complex in animal cell. Of the 16 carbons present in palmitic acid, only 2 carbons come from acetyl coenzyme A and remaining 14 are from malonyl coenzyme A. The main source of NADPH for fatty acid biosynthesis is HMP shunt, hexose monophosphate shunt pathway. When fatty acids are the principal fuel supporting the whole body metabolism, ketone bodies are produced in liver. So ketone bodies are mainly produced in liver mitochondria and are exported to extrahepatic tissue. As fuel. Number 3. Acetoacetate and beta hydroxy butyrate serve as fuel molecules in extrahepatic tissue. Acetone is a volatile ketone body and excreted by lungs. Increased ketone body production in hepatic mitochondria is called ketosis. Ketosis is characterized by ketonemia and ketonuria. Ketosis is caused by increased fatty acid oxidation. Ketosis is mild in starvation but severe in diabetes mellitus. Ketone bodies in urine are identified by Rothera's test. Alpha ketoglutarate accepts amino group from amino acids to become L-glutamate. Pyridoxal phosphate is the prosthetic group that found in active site of transaminases. In transamination reaction, alpha ketoglutarate serve as amino group donor. In transamination reactions, amino groups of amino acids are collected in the form of L-glutamate. Glutamine is the non-toxic transport form of ammonia. Ammonia transports from skeletal muscles to blood in non-toxic form called L-alanine. Transamination followed by oxidative deamination is called as transdeamination. Dehydratase act on hydroxyl amino acids. While desulfide-hydratases act on cysteine. After the loss of amino group, the remaining carbon skeletons of amino acids are called alpha keto acids. The carbon skeleton of amino acid becomes alpha keto acid in transamination reaction. Leucine and lysine are purely ketogenic amino acids. The branch chain amino acids are not degraded in liver but degraded in extrahepatic tissue. The carbon skeleton of asparagine and asparagine and aspartate are degraded to oxaloacetate. Liver lacks the enzyme branched chain aminotransferase, for which branch chain amino acids are not degraded in liver. Oxidation and phosphorylation are coupled by a proton gradient. The energy coupling mechanism is called chemiosmosis, that couples electron transport chain to ATP synthesis. The current mechanism of ATP synthesis is based on chemiosmotic theory and introduced by Peter Mitchell. Coupling of proton flow to phosphorylation of ADP is catalyzed by increasing redox potentials. The ratio of ATP synthesized for half O2 reduced to water is called as P/O ratio. Intracellular ADP and mass action ratio are two factors that regulate oxidative phosphorylation. Oxidation and phosphorylation are coupled by proton gradient. It's the same question repeat ହେଇଛି. Which mitochondrial enzyme complex is associated with ATP synthase activity? Complex 5. World's smallest molecular motor, ATP synthase. What is the P/O ratio? Ratio of ATP synthesized for half oxygen reduced to water. What are the P/O ratio for oxidizing 1 NADH? 2.5 for NADH. Then FADH 1.5. So because P/O ratio depends on the number of C subunit in the F0 complex, which ranges from 8 to 15, P/O ratio varies among species. The two half reaction of an oxidation reduction are linked by electron transfers. Electron carriers of ETC are sequentially arranged in order of their increasing redox potential. मने पका आमे कहितले नेगेटिव पोटेंशियल टू पॉजिटिव पोटेंशियल. Each molecule of NADH carries a hydride ion which bears 2 electrons and 1 proton. Peter Mitchell proposed in 1961 that electron transport and ATP synthesis are coupled by a proton gradient, which is the collection point for electrons between NADH and FADH2, coenzyme Q. Respiration linked phosphorylation is catalyzed by ATP synthase. Oxidation and phosphorylation are coupled by ATP synthase. The two functional domains of ATP synthase are FO and F1. The FO-F1 complex catalyzes ATP synthesis, while isolated FO unit catalyzes ATP hydrolysis. सांग्रे रहिबे हेले सिंथेसिस करिवे, आइसोलेट हे गले हाइड्रोलिसिस करिवे. Then dash and dash maintain structural integrity of respirosomes. Cardiolipin and auxiliary proteins. Cardiolipins and auxiliary proteins. तो बायोकेमिस्ट्री उपरे एतकी क्वेश्चन अच्छी एतकी क्वेश्चन को बोल के रिवाइज कर दबो और जहाँ प्रीवियस क्वेश्चन आम सब टेस्ट करते रिवाइज कर थैंक यू।